going to spend much time talking about Grayson's abilities as a researcher because you're going to hear about it. And I also like to say that every time I've had a graduate student, I've learned a lot. And that's basically what the experience is. They bring more to you that often you give back. And in terms of Grayson, it was a lot about physiology that came with a passion for insect physiology. I tried as much, not being a physiologist, to facilitate his ability to follow up on that. But another thing that Grayson did in this department was really build the department. He really was a champion of graduate students and undergraduate students. He was able to make a tremendous impact. He mentored 14 undergraduate students across various majors, including entomology, ecology and evolution, forensics. I mean, he's really been sort of working across with a lot of students. He's received, as a result, a Graduate Student Impact Award 3 consecutive times from the Undergraduate Club in Entomology. He's been a leader in the Graduate Entomology Student Association. He's organized meetings, meets and greets, outreach events, always trying to champion entomology, the field we love, and try to pass that on to the rest of the world. Because as we know, if the rest of the world doesn't wake up, we're all dead. So it's really important that we have people like Grayson to continue this tradition. And Grayson, go for it.
Oh, thank you. That's very nice. I'm not gonna spend a lot of time introducing myself. I think everyone knows me, but thanks for coming. I know some of you have actually flown in. It's very nice of you. Today, talk about the work I've done for my dissertation. And it all sort of ties around mosquito behavior, specifically blood feeding and what drives it. So just as a bit of overview of the chapters we're going over. So this is basically my dissertation. And we're going to start with just a little bit of background about the internal and external factors that drive this behavior, which we study. So the first question we need to ask is, why blood feed? Why does a mosquito need blood? Primarily, it's for egg development. So if you don't know, only female mosquitoes blood feed. Males and then females, when they aren't developing eggs, feed on nectar. But this behavior itself is pretty interesting because it puts the mosquito at high risk, right? They have to find a host and remain stationary on it while they feed. It's energetically expensive both to find certain hosts and select them in an environment. Then also, once they take the blood, they have to process it. And there's compounds in the blood which are actually somewhat harmful to the mosquito. So that digestive process takes a lot of energy. And then once you have a blood meal, it really reduces your ability to fly. So you can see this image here is a mosquito that's taken a blood meal and flying. That blood meal makes up a good proportion of the mass of the mosquito. So they're actually less able to get around until that blood meal is digested. But primarily, the blood is actually just for egg development. It's nutrition that the mosquito can't get from other food sources, carbohydrate-rich food sources such as nectar, other fluids like that. And the components of the blood that are important are these macronutrients, things like lipids, proteins, but also micronutrients, things like iron and cholesterol have a pretty big effect on the total reproductive output of a mosquito. So if anything of these things are deficient, it has a negative effect on the mosquito itself. So what factors shape blood feeding? So this is kind of a complicated image. It's from my review. If you want to read more about this in general, there's a citation there. But just to go over some of these things, and I'll go over a few more in greater detail, all of these things work in tandem and shape this behavior that we are all very familiar with, right? So broadly, you can divide them into external and internal factors. So things are outside and inside the mosquito. Externally, things like competition. So if they're growing up in an environment with a lot of other larvae or, we're all competing for the same food resources, that affects adult size. In turn, that affects their behavior, as well as things like their total or body size and longevity. Things like environmental conditions, temperature, rainfall, wind, humidity, also all affect this behavior. They'll either stop blood feeding in adverse conditions or, some conditions are more suitable for them to be active in. Time of day, mosquitoes do have a certain periodicity depending on the species, but also where you are in the world. Mating, so there's actually contributions from male mosquitoes that they pass on to females, which can shape this behavior. At the bottom here, you have the vector triangle. You'll hear a lot of talks and papers talk about this, which is the interaction between mosquitoes, which spread disease or spread pathogens, interactions between the hosts they feed on, the interactions between the hosts and the pathogens, and then also the pathogens and the mosquitoes themselves. And then there's also this interface between the external environment and how it becomes perceived and affects things internally. So things like stimuli that the mosquito receives, nutrition, their experiences, these are all sensed and internalized, and that in turn affects things like hormones, the sensory systems, how they find things, the neural architecture, higher order processing of that information. And that can be turned into associative learning and experience. And then all those internal factors manifest themselves in what we can see externally through things like choices, their activity, and infection status. So some of the internal factors, if we take a look inside the mosquito, right? That was a cool animation. That's worth a PhD. So the regulation of behavior through these physiological states, right? These sort of distinct periods of mosquitoes' blood feeding cycles, it prevents them from taking blood when not needed, right? If they can't utilize it for eggs, then they don't take the time to actually feed on blood, right? So associated with these are patterns of gene expression, hormone production, and neurological activity in two very broad states. So there's a pre-blood meal state before they've taken blood, which they are primarily tuned for host acquisition, right? They're sensitive to host stimuli. They're ready to find and take a blood meal. And then after they take a blood meal, a lot of those systems turn off. There's an inhibition of host seeking as these eggs begin to develop, and then they find a place to lay them. So another aspect of the internal side of things is the hormonal control of blood feeding, something I'm quite interested in. But these blood feeding cycles occur in 3 stages, right? Pre-vitellogenic egg development. So once a mosquito emerges from the pupae, there's a period of about 48 hours in which they are not actively looking for a blood meal, and the eggs actually develop to a point where they can receive the nutrients from the blood meal and utilize them. So after that 48 hours, the eggs stop developing, the ovaries stop developing, and that state of arrest. And this is the time in which they're actively looking for hosts to take that blood meal. And once a blood meal is taken, the vitellogenic stage begins, and that blood is utilized to produce yolk proteins, and it's deposited inside the eggs. Then the eggs are laid, and then the cycle begins again. So the hormone that seems to control this is juvenile hormone. I'm sure a lot of us are familiar with juvenile hormone, especially if you've listened to me talk about anything. This is a conserved hormone across insects. So all insects produce juvenile hormone. And in immature insects, it regulates development and maturation, where if juvenile hormone is present, it prevents them from reaching adulthood. And then once they're ready to molt to adulthood, juvenile hormone is no longer produced. But in adult mosquitoes, it's co-opted to have a new function regulating these cycles of blood feeding. So higher titers of juvenile hormone are associated with active host seeking and blood feeding. You can see here on the graph these stages I just talked about, but once a mosquito emerges, there's a spike in juvenile hormone in the black line as the pre-vitellogenic egg development occurs. And then during the state of arrest, it's produced at a pretty constant level until a blood meal is taken, right? And then juvenile hormone drops off. They're no longer actively looking for hosts until they deposit those eggs and they're ready to take another blood meal. And so we can also look at the sensory systems inside a mosquito to determine, to look at some of the things that shape their behavior. Mosquitoes actually integrate a lot of different stimuli in terms of assessing and finding hosts. Things like smell, sight, taste, heat are all stimuli. So the cues that a mosquito responds to are variable between different species of mosquitoes, but also the hosts themselves produce different cues that a mosquito can respond to. At the top right, you see these 2 images. These are 2 species of mosquito, Anopheles gambiae and Aedes aegypti. And you can see the attractants that a person produces. Some are shared, right, that are attractive to the mosquitoes, like CO2 and ammonia. But there are also some that are that each species responds to specifically. So these are, you can look at what a person produces, and depending on the species, what they respond to. These receptors are in the antennae primarily. They're tuned to specific odorants. And the combinations of these odorants are how we are assessed by mosquitoes. And then once they're closer to the host, they will actually integrate different cues. Olfaction, sort of a long-range stimuli. So things like vibrations, heat, color, tastings that are on the surface of their host. So you can see here, these are the sort of sensory apparatus of a mosquito, the antenna, the sensilla vascus, the palps, as well as the eye. And then this is the range at which these things are produced, in which range the mosquito can sense them. So outside of the mosquito, one of the things I want to talk about a bit is host preference and availability. So mosquitoes show some preference towards different hosts, usually in kind of broad groups, like a bird feeding mosquito. They'll feed on certain birds rather than other organisms. There's some mosquitoes that feed on people, preferably. They're anthropophilic. And we can determine this through lab assays. You give them a choice. They'll reliably choose between one or the other, or taking mosquitoes that are fed on blood outside and then analyzing it to determine what species they've been fed on. The host use can vary seasonally, so depending on the time of year. One of my chapters talks about this shift in the late fall, or the late summer and early fall, but these shifts can be important from an epidemiological standpoint, as if it's a mosquito that feeds normally on a bird, for example, starts feeding on humans, that's an opportunity for transmission of disease from a reservoir to the human hosts. So, big question always to ask yourself is, who cares? What's the point of studying these things? So there's two models here, which both estimate the risk of a mosquito to spread a pathogen. On the left is an older model, this model of vectorial capacity. The main variable I want to pay attention to is A. So A is the biting rate, human biting rate of a given species. Typically, this is considered fixed. You say this species has this value for likelihood to bite people, but that doesn't really account for any changes in behavior. It's assuming that they will do this all the time consistently. Another model which sort of accounts for changing behavior is Kilpatrick's model, which is transmission by a given mosquito species. West Nile virus was the focus of this paper. You have all the variables here, but the one that's important to us is this FM. So it's the fraction of blood meals that are derived from mammals, in this case, us. And so this model can account for changing mosquito behavior by looking at those fractional blood meals over time and location. We can estimate how likely they are to feed on a person at different times. So what factors shape blood feeding? We just talked a little bit in detail about all of them, but it is a pretty complicated thing. We're going to be talking today primarily about this area here, the vector triangle, the interaction between mosquitoes and hosts, some chapter about seasonality, and then finally we'll stop off with some hormonal control of blood feeding. So getting into the research now, first chapter is actually a pretty interesting project with collaboration with Jim Simon's lab in the Department of Plant Biology, looking at catnip as a cultivar for repellents. So just a bit of introduction. When I started, about a month after, I got an email from a friend, Ivy, who sent me just a short email. Interesting read with all your favorites, cat emoji, mosquito emoji. And she sent me this paper. It's a really interesting paper about the compounds that are attractive to mosquitoes, or attractive to cats, actually repel mosquitoes. So by interacting with catnip, cats gain some resistance to mosquitoes, sort of serves as a repellent. It's perfect for me. I work with mosquitoes. I also have two cats here, Mimi and Goma. So I talked to Dina about it. I was like, hey, did you know this is a really interesting paper? And she's like, yeah, I know about it. There's a whole project you can work on if you'd like to. So using catnip as a repellent, it's very promising. These bioactive components of catnip, nepetalactones, are thought to could be used as mosquito repellents. There has been some resistance to permethrin and DEET in some mosquito species has been observed, right? So these two primary, very widely available repellents. So it's always good to have more options in the case of these things happening. But what's limited the development of catnip-based repellents in the past is actually characteristics of the plant, right? So how the plant grows and the amount that of these compounds it produces has sort of limited us from being able to utilize it as an effective repellent. So going back to this image, thinking about the vector triangle in sort of this, in using repellents in a broad way, we can actually prevent this interaction, right? So if you keep mosquitoes from interacting with you, ideally preventing when they probe you, as that's the opportunity for pathogens to be transmitted, right? You actually you prevent disease transmission, of course, but so you're altering the interaction between people and the pathogens. And then in turn, in cases where humans are we both get the disease, but our reservoirs and mosquitoes have to pick it up from us again, things like malaria and dengue. If you use repellents widely enough, right, you could potentially prevent mosquitoes from becoming reinfected. So besides the obvious benefits of like, oh, you don't want to get bit by mosquitoes, there's actually a very kind of interesting epidemiological effect of using them in a broad way. So the two catnip cultivars we're looking at were developed here at Rutgers, CR9 and CR3. They were developed here, and the characteristics of the plant themselves make them more suitable for using or to develop repellents. So these have high yields of essential oils, so the parts of the plant that contains the nepetalactones, those repellent compounds. They're also winter tolerant. They can be grown up here in the Northeast, and then they're harvestable using machines, so more effective to use. Also, other work in this department has shown that the CR9 essential oils are repellent to bed bugs and ticks, so two other blood-feeding arthropods. And so my work is testing this on mosquitoes. Do these products or these cultivars can use them to develop mosquito repellent compounds? So the objectives were to figure out, can we use these cultivars to make mosquito repellents with mosquitoes? And then I wanted to answer it in two ways. What's the minimum effective concentration? So it's like the least amount you would need to apply to yourself for the lowest concentration of those repellent compounds that have repellents against common mosquitoes. Do extracts and oils perform similarly? So we have two ways of preparing these products. And is the effect specific to mosquito species, right? If you have a repellent, you don't want it to only repel some mosquitoes, you want to repel all mosquitoes. And then the next question, how long does this repellency last after application? So, if you apply it to yourself, how long are you actually protected? So to test this, we use two species of mosquitoes, both vectors of disease, so Aedes aegypti and Anopheles quadrimaculatus. Aedes aegypti is a globally invasive vector, primarily of a lot of arboviruses. And Anopheles quadrimaculatus is our native malaria vector, so it's sort of a stand-in for a lot of the malaria mosquitoes across the globe. We purchased our mosquitoes when they arrived. They were between 4 and 8 days old. Then we placed them in our incubators at standard conditions, deprive them of sugar, make them more likely to want to bite for 24 hours. And then we began our experiments. So talking a little bit about the things we're testing, again, this is a collaboration with Dr. Jim Simon's lab. We have these oils and extracts I'll talk about for the rest of this talk. The oils have a higher concentration of nepetalactone, those repellent compounds, but a lower yield. It takes a lot of plant to get this amount of oil. So we also wanted to see the extracts. So they have a lower overall nepetalactone concentration, but it's a higher yield per amount of plant material you add. And you can see here it's very green. It has a lot of plant material in it. So there's other compounds present. We were interested to see if maybe some of those extra compounds could increase the repellency if they have their own effect too. So for the first experiment, just laying it out. So we had these treated filter papers. So we treated them with whatever the repellent was, and then place them under a heat pack. So that heat pack is attractive to the mosquitoes. They'll try and interact with it as if it were a host. You can watch them probe it. We tested CR9-3 and CR9-CR3 oils and extracts, and we compared them to DEET, sort of a standard repellent, gold standard, and an untreated control. So we used 400 microliters of each treatment applied to the filter paper in 0.5, 5%, and 10% concentrations. And then our untreated control was that ethanol treated filter paper. That's what the other products were dissolved in. We performed our experiments in our insectary in a HEPA filtered fume hood. And what we did was we filmed from below the filter papers and that are on the heat pack. So you can see here very faintly, this is the treated filter paper. This was an extract treatment group. You can see this line of green, and you can see the mosquitoes interacting with it. So they try and probe through that. We counted the number of landings over 5 minutes. We used 10 mosquitoes per replicate and then 4 replicates per treatment. We randomized the order of the treatments and tested them over 2 days. So we used a random number generator to determine the order of our testing of these different groups. And we did a Kruskal-Wallis test across the treatments to determine significance, and then a Dunn's test for pairwise comparisons. And what we found is that it works for Aedes aegypti for sure. So it has a statistically significant effect. If we look at the graphs here, each is a different product being tested. So DEET, for example, is at the top left, CR9 extracts at the top or at the bottom right. And then on each of these graphs, we included DEET 10% just as a visual comparison. And what we found is that the 5 and 10% treatments are statistically significant. They do have a significant reduction in the number of landings, but not the lowest concentration for any of them. So the 0.5%, you can see this AB, not statistically different from the controls. And looking at Anopheles quadrimaculatus, our other species, we focused primarily on the CR9 products because in our last set of experiments, we didn't see a difference between CR9 and CR3. We found that all treatments reduced landing significantly. So in this case, even at the lowest concentration, we had a significant reduction in the number of landings. So there's no difference between the concentrations of the products or the formulations themselves. So next set of experiments, we wanted to do a time trial. So the setup's identical to the prior objective, right? We had these cages, this heat pack and treated filter papers. But what we did is we treated the filter papers prior to when we began the experiment. So we would treat them, say 12 hours before the experiment begins, so we know how long the repellent has been applied to see if that protection occurs over a certain time window. So for 80s, we tested filter papers that were aged for 1 hour, 6 hours, and 12 hours prior to beginning the experiment. We tested 10% CR9 oil and DEET. And then in Anopheles, we extended that time out to 6 hours, 12 hours, and 18 hours. We looked at CR9 oil and extract, as well as DEET, everything at 10%. So for Aedes aegypti, we found that there was, again, a statistically significant lower number of landings, but also no difference between treatments or time points. So you can look at the graph here. You see 0 landings really across all the treatments, complete protection at any time point, as well as between DEET and CR9. There are not a lot of statistics needed to tell that this is a pretty big result. For Anopheles, again, we also found a significant effect across all time points, as well as formulations, so no difference between the extracts or the oils. However, we did see that higher variability in the 2 later time points of CR9 extract. So we did see some landings, but not enough for it to be statistically different from the treated control. So a little bit higher variation in response to the extracts. So in conclusion for this chapter, right, CR9 and CR3 do provide protection similar to DEET. In our study, we found 93 to 100% protection in terms of reduction of landings. We found that CR9 repels both Aedes aegypti and Anopheles quadrimaculatus equally well. 10% CR9 oil and extract are comparable to DEET up to 18 hours. So we found a really long-lasting protection, but did not find the sort of endpoint where this protection stops. And then, from this result, we can determine that, CR9 and CR3 products are worth exploring in the future as promising alternatives to available products, right? But I'd like to do more studies in a more applied setting, right? You apply these things to a person, may have a different effect on the amount of protection you get, things like your body heat, your sweating. You have like a microbiome, may reduce the time of protection. We'd like to figure out that drop-off point, but I'm pretty happy with these results so far. Have a drink, right? So in the next chapter, I want to talk about is the effects of age and photoperiod on the transcriptome of Culex pipiens. This is a project when I met with Dina for the first time, she told me about this system. I was like, okay, I want to work on that. That's pretty cool. So really the thing I've been working on the longest. So just a bit of introduction about this species of mosquito itself. Culex pipiens is another globally distributed mosquito vector in temperate areas of the world. You can see on the map at the bottom right here, the green is all the places you can find Culex pipiens, right? So just all over the place. It's important to us, in the Northeast, it's the primary vector of West Nile virus. So this mosquito transmits West Nile virus to people, but it's also a competent vector of other pathogens. If you look at the things this mosquito feeds on, primarily it's a bird-biting mosquito. It's strongly ornithophilic. Avian hosts can make up to 99% of the blood meals observed in field-caught mosquitoes. This is important because birds are the reservoir for West Nile virus. So it's a bird disease that these mosquitoes will pick up and then transfer to us. So it's kind of a bit of a conundrum here, right? So if they're transmitting West Nile virus to people, right, but they're a bird-biting mosquito, like how does that occur, right? This doesn't really match up. So that's actually what we're looking at is this very interesting period of the year. So in the fall, the proportion of blood meals that Culex pipiens will take from mammals increases. So if you look at this graph, what you can see here, this gray line is the proportion of mammalian blood meals, so how often they will feed on mammals. The blue bars are the number of Culex pipiens you catch in a trap night. So you can see in the summer, it peaks, and then as it gets colder, it starts to drop off. And then the red is the proportion of West Nile virus cases in the year. So there's a period of peak West Nile virus cases right here. You can see this is time, weeks on the x-axis. And so we look at this time period, kind of interesting, right here. When they're feeding on mammals, they're more likely to feed on mammals. It also coincides with periods of peak West Nile virus production, but also a decrease in the number of mosquitoes you're catching. So even as the populations of Culex pipiens are going down, you do find that they are more likely to feed on mammals. So this is supposed to, or thought to be, due to the migration of their preferred host, American robins. But I wanted to look at it from a different perspective. Maybe something's going on in the mosquito itself that is causing this shift in behavior. So at the same time that all this is occurring, something interesting also is happening. These mosquitoes are going into diapause, so they're getting ready for the winter. So during the late summer and fall, newly emerged mosquitoes, so ones that emerge from their pupae, will start to prepare for diapause. This occurs when day lengths drop below a certain number of hours. The late instar larvae and the pupae are capable of perceiving that difference in day length, signaling the onset of winter as the days get shorter. And then once they emerge, they won't feed on blood. Instead, they'll prefer to feed on carbohydrate-rich food sources. They accumulate a lot of fat. They don't develop their eggs, and then they overwinter as adults. So they are behaviorally very different. Also, visibly, they're actually quite different. You can see at the bottom here, these are the Culex pipiens. They have this big reserve of fat, and they are unresponsive to host. So with that in mind, we can make some assumptions about the population of Culex pipiens at this time of year, right? We'll assume that they're older, because if you were catching biting mosquitoes at this time, it means they've emerged prior to that photoperiod cutoff, but the adults themselves are still exposed to diapause signals, right? They can still perceive that shorter photoperiod, even though they've kind of missed the boat to enter diapause themselves. So my questions were, do these factors, age and photoperiod, do they affect the gene expression of adult female mosquitoes? And do changes in these genes maybe explain some of the differences in host use that we observe over the season? So to test this, we collected Culex pipiens from behind CVB, very common mosquito. We collect the egg grafts and reared them under these long day conditions. So these non-diapause inducing photoperiods. We collected them over 2 summers, 2022 and 2024, one to make up for some of the quality of the libraries I made, and then also to account for maybe there's some differences between the seasons. So we'll combine them and hopefully address some of that variability. Following emergence from the pupae, we had 2 conditions we transferred the adults into, right? So we either kept them in the same long day length, non-photoperiod inducing condition of 16 hours light, 8 hours dark, or we moved the adults into a incubator at a diapause inducing photoperiod. So if they were reared as larvae under this condition, they would definitely enter diapause of 12 hours light and 12 hours dark. Kept them sort of normal conditions and gave them 10% sugar to feed on. And then over the course of about a month, so 4 weeks, we collected mosquitoes from both groups at 1-week intervals for RNA extraction. So, 5 to 7 days up to 26 to 28 days. We had the groups up here, the sort of experimental things we're looking at are the long day length group up here, the short day length, right? And so a group of mosquitoes from each time point in each photoperiod. We extracted RNA with pools, 4 pools of 3 mosquitoes per treatment group. I chose to pool them, hopefully to account for some variation between individuals that would occur in gene expression, and then sequence them using Illumina HiSeq. We then took our RNA reads, performed some QA/QC, and aligned them to the Culex quinquefasciatus genome. We could then able to generate count data, so how many times a segment would align and count within a certain gene, and then did differential expression analysis using DSeq, so determining the actual differential expression between genes. And so the comparisons we're interested in, so I wanted to see the effects of photoperiod, so we compared long day and short day within the same age group. So, compared the week 1 old long day to the week 1 old short day for each time point. And for the age comparisons, we compared each treatment group to week 1 within the same photoperiod. So we kept all of our comparisons within either the short or the long day, and use week 1 as sort of a standard. You find in a lot of blood feeding studies, 5 to 7 days old is like prime biting time. So we wanted to compare everything to week 1 and see how that changes as the mosquito ages. We considered our genes differentially expressed if DC determined they had a adjusted p-value of 0.5 and a log fold change value of plus or minus 1. So either downregulated or upregulated by a factor of 2. And then once we had these genes, we then assign them Go terms. So if you're unfamiliar with what a Go term is, it's just a way of categorizing a gene based on its function or where it is. You have these 3 broad categories: cellular components, so where in a cell it's expressed; the molecular function, what reaction it's actually involved in; and then biological process, sort of like a bigger picture thing that these genes are involved in. So we used vector base for this to determine which Go terms were statistically different between our comparisons. So, we had 10 comparisons and, like hundreds of genes. So I can't talk about all of them. I want to talk about the ones I thought were most interesting, starting with the photoperiod comparisons. So across the photoperiod comparisons, we found a lot of differentially expressed genes that were pretty interesting. Some include trypsins and peptidases. These are proteins that are involved in breaking down of other proteins, as well as trypsin is involved in the digestion of blood. So these mosquitoes weren't blood fed, but we did find differential expression of trypsins. And then farnesyl dehydrogenase and Kupel homolog are both juvenile hormone related genes. You can see here in this pathway, this is the JH synthesis pathway. Farnesyl dehydrogenase is the first step in the JH specific steps of this pathway. And Kupel homolog is a transcription factor that's in a lot of other insects is involved in different processes related to juvenile hormone. Looking at the Go terms, we found a few that statistically different Go terms that were interesting: carbohydrate metabolic process, heme binding, and egg chorion. And I thought that was pretty interesting to see, because what we found is that some of these differentially expressed genes and processes, like those Go terms I just talked about, are actually associated with diapause. So while these aren't diapausing mosquitoes, they weren't, didn't accumulate a lot of fat, they weren't reared in a condition that would induce diapause, we did find that some of these genes were differentially expressed. So non-diapausing mosquitoes are affected by the shorter day length. And this in turn could affect survival, different aspects of blood feeding behavior, as well as the digestion of blood. So all things that are relevant epidemiologically. And so moving on to the age comparisons, one gene that was really interesting that turned out was sarcoplasmic calcium binding protein 1. This wasn't necessarily something that explains the difference in host use, but we found it differentially expressed across all of our age comparisons except for one. And it's interesting because the homologs of this gene are used to age 80s mosquitoes. So people have used them to estimate the age of mosquitoes using expression of this gene. So you can see here, this is a graph from a paper where they do just that. You can see mosquito age and then normalized gene expression of this protein over time. You're able to use it as a tool to estimate mosquito age. So going forward, I'd like to actually investigate this more in QLEX, which hasn't been done yet. Just an interesting finding. Also makes sense, good to kind of confirms, there is a difference in age, but also you would expect this this protein to be differentially expressed. So this is a nice thing to find. Some other things we found were a lot of genes that are involved in muscle function or formation. So you can see some of the proteins here that are involved, but like troponin, tropomyosin, myosin regulatory light chain, the Go terms, contractile fiber, myofilament, myofibril. These are all things that have to do with muscle function. And these tend to be downregulated as we look at older mosquitoes. And what we thought is maybe that this decreased muscle function, right? They're not, the muscles aren't functioning as well, may affect their ability to actually reach birds at the tree height. So you'll capture more QLEX pipiens in traps that are placed higher up where birds are. But as the mosquitoes get older, right, or as the population of mosquitoes gets older, they might just not be able to reach that height. And incidentally will occur more mammals, could explain why they're feeding on mammals more often at this time of year. And then finally, we found a lot of odorant binding proteins that were upregulated in the different ages compared to week 1, except for 2. Odorant binding protein 56A and 66 were downregulated at week 4. I tried to figure out, has there been any research into what these things actually bind to, what they are responsible for in terms of the mosquito, what they can sense, and it's not really known. So we don't know what these the function exactly of these odorant binding proteins are. But having differential expression suggests that there may be some differences in host perception, which may in turn explain some of these differences in behavior we observe in the late fall or late summer, early fall. And so in conclusion for this section, both age and photoperiod were found to have a significant effect on adult Culex pipiens gene expression. Seasonal differences in this host use may be due to differences in physiology, right? So instead of just a difference that is based on the abundance of the host, right? The hosts are migrating. We're showing that these mosquitoes and their these two different factors could explain, at least partially, some of these differences we see in behavior. And then finally, one thing we found and is unexpected and pretty interesting is maybe receptivity to diapause signals in Culex pipiens isn't as discrete as previously thought. Maybe, while we historically have looked at diapause in this mosquito as something that's actually very visually easy to observe, right, as they accumulate fat and their eggs don't develop, we are finding that some of these processes on a gene level seem to be occurring in adults that are exposed to short day length. So diapause might not be, or may be initiated in larvae and pupae in the strict sense, but adults still respond in some capacity to these short day lengths, which in turn may affect their behavior. Right, so the last thing I want to talk about, last chapter, is about juvenile hormone, the effect of analogs on behavior itself. So as a bit of background, again, we talked about this in the beginning, but juvenile hormone in immature mosquitoes regulates pupation and adult maturation into adulthood. But in adults, it's associated with egg development, early egg development, and host-seeking behavior. So back to the similar version of the graph I showed earlier, right? Once the mosquito emerges from a pupae, there's some early egg development in which they're non-responsive to host as those eggs develop. So they're ready to actually take blood. And then once the eggs are ready, they produce it at pretty consistent levels. And then that drops off once they've taken a blood meal. They become unresponsive to hosts, and they digest and utilize that blood for egg development. And once they lay those eggs, the cycle begins again, right? They'll look for another blood meal to lay another batch of eggs. So importantly, right after the blood meal, this juvenile hormone production ceases. They become unresponsive to hosts. But juvenile hormone is also interesting from another perspective. We utilize analogs of juvenile hormone as larvicides in mosquito control, as insect growth regulators. So things like pyriproxyfen, hydroprene, and methoprene are applied to the environment they cause mortality in the larvae by disrupting development and pupation. So at the bottom here, you have, this is like a larval pupal hybrid, a really strange thing you wouldn't normally see. It's because this larvae had been exposed to juvenile hormone, so that will die. And then this pupae had a failed molt, right? So this adult is dead. And we utilize the action of juvenile hormone as larvicide. So, with that in mind, juvenile hormone seems to have a big role in blood feeding, but we're also applying juvenile hormone related compounds, juvenile hormone to the environment. Adults may encounter these things either by touching bodies of water that are treated or interacting with really anything that may have juvenile hormone analogs in the environment. And because juvenile hormone has such an important role in blood feeding cycles, we thought maybe it will cause strange behaviors in the mosquito, which in turn could increase disease transmission, right? Anytime a mosquito is biting more often, it's a bad thing for us. So my question was, does exposure to these juvenile hormone analogs cause non-biting mosquitoes to blood feed? So to test this, we purchased Culex quincefasciatus from Benzon Research. We gave them blood 24 hours after arrival. So once they arrived, I gave them a period to adjust after shipping, gave them some blood, and then allowed them time to develop those eggs. So 48 to 72 hours, kept them at those standard conditions. And then we had 3 experimental groups. So we had untreated adults. So these are gravid mosquitoes that nothing was applied to. We had a group of mosquitoes where we applied a solvent control. So this is 50% ethanol and 50% acetone. And that solvent was what we dissolved our S-hydroprene, our juvenile hormone analog in. So we also treated mosquitoes with this 2.5 nanograms of a juvenile hormone analog. And so 24 hours after we applied either the solvent control or the juvenile hormone analog to the mosquitoes, we expose them to our blood feeding system. So basically what this is a double jacketed water vessel. So in the internal chamber, you have something that will, it holds blood, which is kept in place by a membrane. And then this is hooked up to a recirculating water bath, which heats the blood so that the mosquitoes become attracted to it. So we expose them to this blood. We gave them an hour to interact with it and feed. And then we counted the number of gravid engorged mosquitoes. So gravid mosquitoes, which had taken a blood meal. We used a chi-square test between treatments to determine the significance, and then did Fisher's exact test for the pairwise comparisons. And we performed this experiment twice, once with randomized treatment groups. So we just did them in a randomized order, either the controls, untreated control, the solvent control, or the S-hydroprene. Then we repeated the experiment again where we did our treatment groups all in at the same time. So we did all of our controls on one day, all of our solvent group on a different day, and then all of our juvenile hormone analogs on a different day. So for our results, I just want to draw your attention up here. So this is what we're actually looking for. So these are the kind of mosquitoes we're working with. So this is what an unfed mosquito looks like. It's what you would expect to bite you in the environment. And then this is what a gravid mosquito looks like. You can see that distension of the abdomen as the eggs are developed. They're ready to lay those eggs. And what we're interested in are these blood-fed gravid mosquitoes. So you can see both the blood here as well as the eggs. Pretty unusual to see in nature for sure. And what we found is that our juvenile hormone analog, S-hydroprene, is statistically significant from both controls, but the controls aren't different from one another. So you can look here at this graph, you see like a pretty significant increase in the number of gravid mosquitoes taking a blood meal. We did see some of both the solvent and the untreated mosquitoes blood feeding, which may be due to some contamination while this experiment's being performed, right? It takes very little of these juvenile hormone analogs to have a biological effect, particularly in larvae. So if this is in the environment at all when we're doing the experiment, it may cause some behavior, even though it's a very small amount. And then for the separate experiments where we performed each of these groups separate, we also included a group of non-gravid mosquitoes, just normal biting mosquitoes, as a comparison to see what rate they bite at compared to our other treatments. And in this time, when we did everything separately, we found that our untreated mosquitoes didn't feed at all, right? So it suggests that, yeah, maybe it was contamination in the last experiment where you had some of these untreated mosquitoes biting, because that's very unusual. Although we did see solvent mosquitoes, the solvent control biting again. This may be due to dehydration. So you can read about it in the review. There are a lot of studies showing that dehydration can cause mosquitoes to compensate for it by taking blood meal. They're more likely to bite. And there's a lot of harsh sort of things in that solvent may have stripped away some parts of the cuticle, dehydrated the mosquitoes. So we think that may have been going on. And then comparing the juvenile hormone group to the non-gravid mosquitoes, they didn't feed at the same rate as mosquitoes that would normally take a blood meal, but we did see, again, an increase that was statistically different from our two controls. And so now we know that there's some sort of effect. I wanted to test the concentrations, change that to see how that affects this sort of strange behavior. So we did the same experiment, same thing where we applied it to the mosquitoes after they had developed eggs, and then exposed them to our blood feeding system. But we changed the amount of S-hydroprene that we applied from 0.25 nanograms all the way up to 250 nanograms. So our last experiment was this concentration. So we had two higher and one lower. We did the same experiment where we provided blood 24 hours after application. But before the experiment, again, I also measured the mortality. So how many mosquitoes died after treatment to determine if there's some sort of a mortality effect as the concentrations increase. We had 3 replicates of about 30 mosquitoes per concentration group. And for this experiment, what we found was there is a response to the different concentrations, with the highest proportion of gravid blood-fed mosquitoes, again, these ones that have eggs and blood, in the 25 and 250 nanogram group. But also we observed some higher mortality in this highest concentration. We didn't have a lot of our mosquitoes not make it that 24 hours after applying that high concentration. So, it's interesting to see that there is an effect, right? We didn't see like the end or the, if this continues to either drop down or go up. But in future studies, right, I'm, still looking at this system. This 25 nanogram seems to be a nice sweet spot for increased of this atypical behavior, but also lower mortality. So the next experiment, applying this stuff to mosquitoes directly is just kind of not like that's not happening in nature. Someone isn't walking around and applying with a micropipette to all these mosquitoes. So I want to see if there's a way to simulate exposure that they would actually experience outdoor. And to do this, we made these little flowers, kind of cute. So these petals are filter paper. We saturated them with a S-hydroprene solution and allowed them to dry. And then we attached them to these wicks that are placed in an Erlenmeyer flask containing sucrose, right? So this sugary liquid they feed on. We had two groups, right? A non-treated control and then petals that had been treated with hydroprene. We placed these in the cages with about 150 mosquitoes per cage, replicated 4 times, using that, again, the untreated flowers as a control. And then after 72 hours of them interacting with this flower, right, after we place it in there, they have a chance to walk over it, to feed on the wick. We counted the number of, again, gravid engorged mosquitoes with eggs, and had taken a blood meal. And we used Fisher's exact test to compare between these two treatments, the proportion of blood-fed females. And what we found, again, we had a significant increase in the number of blood-feeding mosquitoes in the treated group. We did also see some control mosquitoes were blood feeding, again, maybe due to some concentrate contamination. All this was being done in the same room. But interesting result, they are able to pick this up either through walking on the petals themselves or feeding adjacent to it. And then finally, the last experiment we did is like, okay, so blood's utilized for egg production. What happens to this blood meal if they already have eggs that are developed, right? Can they develop more eggs with this blood? Does it just get excreted? We were unsure. And then also looking at, well, there's gravid mosquitoes that aren't blood feeding that are exposed to juvenile hormone analogs. Does this affect reproduction at all, right? Juvenile hormones in these reproductive cycles, maybe there's an effect. So what we did is we had 3 groups of mosquitoes. We did the previous experiment with the flowers to generate these. We had untreated gravid mosquitoes, right? Not treated with anything, just have eggs. And then 2 groups where we exposed them to juvenile hormone analogs, right? So one group was mosquitoes that were treated but didn't take a second blood meal, and then another group of mosquitoes that was treated and took a second blood meal. So we placed these mosquitoes all in their cages, about 30 each. And then the mosquitoes that took a second blood meal, we want to give them time potentially to digest it and use it to develop eggs. So we gave them 48 hours. And then we gave them a site to lay those eggs at, so a little bit of water. We counted the number of eggs that they laid in each raft. And then after that, we counted the number of eggs that hatched from each raft to determine the hatch rate. I used an ANOVA to compare the egg numbers, and then a chi-square test to look at the proportion of eggs which hatched. And what we found is, yeah, there's a statistically significant difference in the number of eggs between the treatments. So that's this top left here, the number of eggs. So not a difference between untreated mosquitoes and treated mosquitoes which didn't take that second blood meal. But taking a second blood meal did cause a reduction in the number of eggs laid. But a little bit more striking than that is the effect on hatch rates. So we look at the bottom right here, the untreated mosquitoes, about 96% of the eggs hatched. In the treated mosquitoes that took a single blood meal, we had a slight reduction at 90% of the eggs hatching on average per raft. But then once they take that second blood meal, that number drops down to 41%. So there's a real reduction in the number of eggs that are hatching if they take a second blood meal and are treated. We don't really understand why this is occurring. I think this might be due to either them, there's a lot of those compounds which may be harmful in the blood, but also, they have to take energy to deal with that away from egg maintenance. So the really a striking result we weren't expecting to find. So in conclusion, juvenile hormone signaling, right, influences blood feeding behavior. This is the first study to look at the effects of juvenile hormone analogs on adult behavior, but also further support for the role of JH in blood feeding, right? So by using mosquitoes which have stopped producing juvenile hormone and are unresponsive to host, right, they're gravid, and then applying these compounds, we're able to further confirm its role in blood feeding, right? And then the use of these things in the environment may influence pathogen transmission, right, by just increasing the number of biting mosquitoes, right? It's also bad. A mosquito that has eggs is bitten before, which means it's given them an opportunity to take up a pathogen. Although we don't really know to what degree this is happening in nature, we'd have to do more studies for that. And then finally, that second blood meal isn't used to develop eggs, not used for oogenesis, and it seems to actually disrupt normal reproductive output. Something I'd like to look into more, determine exactly what's happening at a gene expression level. So with that, tying this all together, mosquito blood feeding is like really complex, right? There's a lot of factors that are involved in it, shaping this sort of like simple behavior that we think simple behavior that we're all kind of familiar with. But we do have some takeaways from this work, right? The development of repellents provides an effective way to disrupt mosquito behavior, and in turn, maybe disrupt that vector triangle we talked about. And then factors like time of year, photoperiod, and age have an effect on epidemiologically relevant aspects of mosquito behavior, but aren't really included when we traditionally think about models of explaining mosquito behavior. So they should be investigated more. And then finally, juvenile hormone regulates mosquito blood feeding. And the application of juvenile hormones is an interesting system to study this. Like I said, these are mosquitoes, gravid mosquitoes, which are normally inhibited from producing juvenile hormone by the presence of eggs. And so we're able to artificially apply this and created sort of an interesting system. So what's next for me, right? What's going forward, right? Dina told me it's normal to have more questions when you end your PhD than when you started. So I have things I'd like to work on, right? One thing is, how do JH levels differ between autogenous and anautogenous mosquitoes, right? This hormone that's so an anautogenous mosquito, what we're all familiar with, biting. Autogenous mosquitoes are actually capable of producing a batch of eggs without taking a blood meal, right? And so juvenile hormone is so important in regulating this behavior, but also that early egg development. I'm interested to know maybe what's going on in terms of juvenile hormone titers between those two groups of mosquitoes. And then can we estimate the age structure of Culex pipiens at that time of the year? We're interested at late summer, early fall, to confirm that they're actually getting older. We made an assumption that these mosquitoes are getting older because if they're blood feeding, that means they've emerged prior to the diapause cutoff. Maybe we can use that sarcoplasmic calcium binding protein, develop an assay for it so we can estimate the age of mosquito population at that time of year. And then one thing I've started working on, I have some data for, is how does the application of juvenile hormone analogs affect gene expression in gravid females, right? So we have these mosquitoes which are doing something very strange. It also, if they do take a second blood meal, seems to have a negative effect on the reproductive output. But what genes are being turned on by the application of juvenile hormone analogs that are responsible for driving this blood feeding behavior in mosquitoes? So it's something I'm working on, have a little bit of data, but need to really flesh that out. And then with that, I didn't take some time to acknowledge people. I couldn't have done this with I have a ton of support. First, I'd like to acknowledge Dina, my boss. I think she took a risk taking me on. She knew I dropped out of grad school once, but she has always been enthusiastic and encouraging. She's kind of let me do all the things I'm interested in, and is just an inspirational scientist. I'm really lucky I didn't really have to read that much for my qualifying exams, because we're having 3-hour meetings once a week just talking about research. So I've just learned a ton. And then my committee members, Alvaro Toledo, Cesar Rodriguez, and then James Simon. I only had one qualifier when I picked my committee. I asked Dina, I don't want to work with anybody who is going to make this harder than it has to be, and who's hard to work with. And you guys definitely haven't been. You've been incredibly supportive anytime I've needed anything, just also very kind people. And then I want to thank the whole department for, they provided me a lot of opportunities. I've learned a lot from all my teachers here. But I especially want to thank Francine. So Francine has, is the lifeblood of this department, make sure that everyone is getting paid on time. And she helps me with these things that I really struggle with, like filling out timesheets and, things that are very foreign to me. So thank you, Francine. Thank you so much. And I'd like to thank my lab, Dina's lab. I've been really lucky. Everyone I've worked with has been welcoming and very tolerant of my somewhat neurotic behavior, and has never made me feel not welcome. And so I'd like to specifically thank Nicole. Nicole is the backbone of CVB. I've learned so much from her. She's also always been incredibly helpful, is just a wealth of knowledge and positive attitude. She is a really amazing person. And then another special acknowledgement to Francisco and Julia, who I ended up accidentally working with one summer. And it was, spent the whole summer collecting ticks and processing mammals. And it was an extremely fortunate accident. They're wonderful people. And I met them early on in my PhD, and they're so hardworking. It really inspired me to what a scientist should be. So I'd like to thank them, just lovely, lovely people. My family, I'd like to thank my parents, Greg and Susie. who are here in attendance, right? You guys have been incredibly supportive my whole life in terms of my education, and then Garrett as well. You guys also are on the bad end of me, like not responding to phone calls and not visiting enough because I'm doing my PhD. So thank you for all that support. I'd like to thank my grandfather, Richard Tung, who passed away before I finished, but he always used to say, if you get your degree, you'll be the first doctor in the Tung family. So I think I would have made him proud. Just an incredible man. And then my in-laws, John and Julie Hawkins, who've welcomed me into their family, incredibly supportive also during this whole process. Another special shout out to the cats as well. My assistant therapists. And then my friends. So I've been in entomology circles for about 10 years almost now, I think. And I've been able to meet really, really incredible people the entire time. So, but I'd like to give a special acknowledgement to Exant and Alex, who I think are in the chat. They really helped me through sort of a down period in my PhD, made me feel like a really, they treated me like a person and made me feel very, very good about myself. They've always been incredibly kind to me. So thank you guys. And then Carl and Jocelyn, who have assisted me professionally, in addition to being my friends. If anyone took I don't know if anyone in my class was, I TA'd for insect classification. Carl was my TA, so you also have to thank him. I just copied him. But they always, they write me letters of recommendation, just really professional, lovely people. So thank you to them. And then finally, I have to thank Chloe. I couldn't have done this without her support, like in any way. Actually, I wouldn't even have come back to school. She really pushed me. I didn't believe in myself, and she never stopped believing in me. She is the hardest working person I know, just an inspiration as a scientist, also just in going about her life. Really an amazing person. So I'm so grateful to have met you and married you. And thank you so much for all your support. So with that, just this last slide, one thing that's not so great about working with mosquitoes, they're not the best looking insect, but there's a few that